Now, without any further delay, I'd like to turn today's program over to your first presenter, Rob Tarbucks. Rob, the floor is yours.
Thank you. Hi, everybody. My name is Rob Tarbucks. I'm a marketing manager here at Illumina in the systems group representing MiSeq. And it's my great pleasure today to introduce Dr. Andrew Fellows. And what he's going to do today is talk a little bit about his experience with both MiSeq and the recently released TruSeq cancer panel. And Dr. Fellows received his undergraduate training at Canterbury University in Christchurch, New Zealand. He then followed that up with a PhD in pathology at Otago University in 2000. He's been in the field of molecular genetics in New Zealand for over 20 years, with the past 8 as the head of molecular pathology at Canterbury Health Labs, which is based in Christchurch. Following the devastating earthquakes in that town in 2011, Dr. Fellows took up the opportunity to head a transitional research program at the Peter MacCallum Cancer Centre, which is based in Melbourne, Australia. And his focus there is developing molecular diagnostic services based on next-generation sequencing. And with that, I'd like to turn it over to Dr. Fellows, and the floor is yours.
Thank you, Rob. And I'd like to thank Illumina for the opportunity to speak to you all today. So, my lab was a small clinical diagnostic lab. It started out doing familial cancer testing about 12 years ago. We still do that, but in comparison to somatic testing of solid tumours, that side of our business is now small. And what has happened in the last 5 years in molecular genetics, especially in the area of cancer, has been quite remarkable. There's been a revolution in cancer therapy that's brought genetic pathology to the front line of the war on cancer. But only recently have clinical laboratories like ours gained access to the technology that we need to meet this growing demand in tumour profiling. Many cancer centres are now busy working out how to translate these technologies into clinical services that our clinicians will use on a daily basis. So in this presentation, I want to discuss our experience with Illumina's TruSeq custom Amplicon panel and describe how it is allowing us to make this transition. So what are the challenges facing labs like ours? Cancer is one of the leading causes of death in the Western world. The World Health Organization estimates 7.56 million people died from cancer in 2008. That's 14% of deaths worldwide. It states that the global burden of cancer can be reduced by preventing cancer from occurring in the first place, by detecting cancer earlier, and by managing patients with cancer. So, cancer is a genomic disease, and increasingly individualized treatment is having a dramatic impact on patient survival. An important part of that is timely analysis of cancer genomes to make sure that patients gain appropriate therapy in an appropriate time. Next generation sequencing, as we all know, has come to the fore recently. It's driven down the cost of genetic data collection over 1 million-fold over the last few years. And this is imposing a burden on diagnostic laboratories which we're not really faced before. This is a new deal. And so traditional laboratory practices will challenge our ability to deliver next-generation-based sequencing services. An example of that is that we're now going to be performing large-scale quantitative assays with extremely complex workflows and a high potential for contamination. This is something that's been familiar to infectious disease laboratories for several years, but with next-generation sequencing, the workflow is much more complicated. So I just wanted to give you a taste of what is driving the uptake in tumour profiling, and this is a picture from New England Journal of Medicine last year, a review of genomic medicine. And this picture is taken from the BRIM-3 trial of bemorafenib treatment for metastatic melanoma, and it shows a PET scan of a patient before and 2 weeks into treatment with that tyrosine kinase inhibitor. And this works for this patient because they have a mutation in the BRAF oncogene, the V600E mutation. And the red and the green areas show the metabolic activity of tumours, which is vastly decreased as the tyrosine kinase inhibitors had an effect on those tumours. So there are many examples at the moment of drugs with the same efficacy, and here's a short list of them. These are the ones that are currently driving the demand in molecular pathology here at Peter Mac. All these biomarkers, with the exception of KRAS at the bottom, are tyrosine kinases for which specific inhibitors have been developed. And these drugs only work if a specific genotype is present in the patient's gene. There's many other classes of tyrosine kinases and other drugs under development. Over 500 therapies currently targeting over 100 different biomarkers. So this is driving an increase in tumour profiling at Peter Mac, which is shown in this slide. And when the new drugs come along, we feel that we can't cope with the traditional methods such as Sanger sequencing. So we need a new way of performing these tumour profiles. So what are some of the problems with doing tumour profiling in this new environment? We're required to do more with less. We often get limited sample. These are biopsies or resected tumours. These samples are embedded in, or treated with formalin and fixed in paraffin, which degrades the DNA and makes it difficult to analyse. Samples are genetically heterogeneous, so there's a mix of cancer cells and normal cells in any tumour, and this requires that we have either we enrich for the tumour portion of the sample, or we have assays with extremely high sensitivity. And in some tumours, there's natural pigments that can inhibit molecular techniques, further limiting the sensitivity of our methods. And clearly, as more biomarkers are discovered, there's an increasing demand to do more with the one sample. So with traditional testing, we can only do a few rounds of testing. This is serial testing. We look at the most likely genes first, and then eventually the biopsy becomes exhausted. So next generation sequencing technology is a solution to this problem. Next generation sequencing technology will improve tumour profiling by providing a higher sensitivity compared to existing methods. And in a recent paper from Roman Thomas's group in Cologne, they showed that of the state-of-the-art methods, Sanger sequencing and Pyra sequencing and Nextgen sequencing based on the 454 technology, only Nextgen sequencing was able to detect all the mutations present in a cohort of 24 non-small cell lung cancer patients. Nextgen sequencing will allow us to perform parallel testing for multiple targets rather than serial testing. This is a great advantage. Nextgen sequencing will allow us to perform parallel testing of multiple samples. So rather than one patient at a time, we'll be able to do many at a time. And so these are two great advances. And detection of any mutation will be possible in the region of interest rather than using an assay which is targeted to specific mutations such as a genotyping assay. Assays such as the OncoCarta panel from Sequenom have been available for a number of years. They've been a great boon to researchers and to some diagnostic laboratories, but they suffer from the problem that they have a high false negative rate because they only target precise mutations which are known to be present, which are known to drive cancer, and they're not sensitive to mutations outside the region that they're targeted for. Okay, so I just wanted to take you through the evolution in technology that's occurred over the last few years. So our laboratory has survived for a number of years on capillary sequencing. So this is a traditional technology. I've taken sort of an estimate here of the amount of data that you can produce if you drive one of these machines very hard and the cost it would take. And I guess one of the problems here is that it takes quite a few days to produce data. So, 5 to 6 days to produce a relatively modest amount of data in cancer genome terms. At the other end of the scale, we've had available from Illumina now for quite a while the MiSeq 2000. This is the big daddy of genomic sequences. You can produce more than one genome's worth of data, but once again, the timescale is unacceptably long. Although upgrades are coming later this year, which will dramatically improve the time frame. But also the reagent cost in the MiSeq is prohibitive for diagnostic labs. So up until just within the last year, we haven't really had technology in the space that's really well suited for diagnostic labs. But late last year, we got access to MiSeq, the personalized sequencer from Illumina. This is one of a few on the market. MiSeq's pitched really well for diagnostic labs. It's got a good short turnaround time. It produces a moderate amount of data, but enough data for genome analysis of tumours. I've shown here 6 gig of data. This is what MiSeq will be capable of by the end of the year. At the moment, it's more like 2. And so, based on combination of price, run time, and amount of data it produces, we feel MiSeq is well suited to the diagnostic lab. So we've decided to use this instrument as the basis for our tumour profiling experiments. So a little bit about the MiSeq. Got a single flow cell compared to its big brother. Everything's done on board. There's no the generation of the libraries or the so-called cluster generation is all done on the instrument. It's got a very simple interface. Importantly for laboratories, the reagents are tracked by RFID, so there's full control of consumables. Produces a good amount of data and got very good QC parameters, so greater than 75% of bases are passing Q30. And with the upgrade by the end of this year, we'll be producing 2 by 250 base pair reads on this instrument, which is quite impressive for what's traditionally regarded as a short read instrument. So you'll notice Nature Biotech has recently published a paper from Nick Lohmann's group in the UK comparing personalised sequences in this space. I highly recommend you read that paper. It's got some good insights into the strengths and weaknesses of all the personal genomic sequences, and we're really happy that we've got access to the Illumina product, and so I recommend you read that. So obviously for looking at tumours, we need to look with high sensitivity, and I just wanted to talk a little bit about the differences of ways of sequencing genomes. So at the moment, we can classify Nextgen sequencing into wide and deep. And what are the differences between these two things? So wide sequencing I've classified as where you produce a low number of reads across the region of interest, so maybe less than about 50. Whereas deep sequencing I've classified as maybe greater than 1,000 reads per region of interest. Wide sequencing is suitable when you have variants that are expected in high abundance, for example, germline mutations. Whereas deep sequencing is more appropriate when you're expecting to find variants in lower abundance, such as somatic mutations. Wide sequencing generates a very large volume of data, and it's complex to analyze bioinformatically. Whereas deep sequencing, it can generate a large amount of data, but in general it's simpler to analyze. It takes a shorter amount of time. Wide sequencing obviously has the potential to discover variants. It's why it's done. But from the clinical point of view, often these variants are of uncertain significance. Whereas if we do deep sequencing, we can really focus in on the regions that are that we know are likely to contain variants of clinical significance, such as the active sites of tyrosine kinases, and this limits the potential to discover variants of uncertain clinical significance. So, there's several approaches to enriching for targets of interest, and we've chosen at Peter Mac to focus on an Amplicon approach. We took that decision as far back as 2009, and we initiated experiments using microfluidics to create highly targeted Amplicon genomes. But why is Amplicon better than other types of target enrichment? For a diagnostic lab, it's good because it's similar to our existing routine workflow. Diagnostic labs are very familiar with PCR. So in terms of validation and just general laboratory equipment, it fits in very easily. It also allows us easily to create the tags and sequences that are necessary on Amplicons to make them available to work on Nextgen sequences. And Amplicon sequencing really allows us to use a relatively modest amount of input DNA, so it's highly amenable to FFPE material. And finally, we can control very precisely the specificity of the target region using Amplicons. Other approaches, such as capture, have a high probability of giving you off-target reads, and this complicates the bioinformatics, and it's not as desirable. So we prefer, especially when we're looking at genes which have a high number of pseudogenes, such as BRAF, to use a targeted Amplicon approach, and we've proven the success of this strategy. So the Illumina launched the TruSeq, or announced TruSeq custom Amplicon cancer panel earlier this year, and Peter Mac was lucky enough to get access to this product. So I'll just describe it a little bit. It's an off-the-shelf product, so it's been designed in-house. It contains 212 Amplicons. Each Amplicon has been designed to work optimally on FFPE. As you'll know, FFPE material is highly degraded, so choosing Amplicons which are less than 200 base pairs is very important. It's been designed with built-in sample indexing, so you can mix up to 96 samples in the sequencing run and gain information on all those samples simultaneously. It takes approximately 2 days from the beginning of the experiment to starting to get your data to analyze. So you've got 1 day of creating the library and normalizing it, and then 1 day of sequencing on the MiSeq. And it's only available on the MiSeq. This is a product that's designed with that instrument in mind. So the official input material requirement is 250 nanograms. We are trying to push that down, and I'll show you some experiments that discuss that. So we got access to this panel earlier in the year. We ran through 88 clinical samples that were derived from FFP material. We also put on some cell lines that were mixed in known proportion with wild-type DNA to try and assess the sensitivity of this panel. I just want to talk a little bit about this product. It's a research use only product in the United States. So in Australia, or here at Peter Mac, we're using it in our laboratory, which is a NATA accredited laboratory, equivalent to CLIA. But the intended service for this product remains as a research one. We're providing it to one research client only, which is the Cancer 2015 project, which I'll talk about later on. So we just have to bear that in mind, that it remains a research product. So now, it's important to understand the relationship between batch size and library size and sequencing depth on an instrument like MiSeq. So you can choose any number of samples and any number of Amplicons up to the limit of the assay. but you'll notice an effect on the read depth that you achieve per sample or per amplicon. So here, for example, I've shown if you have 12 samples in your MiSeq run and only, say, 48 amplicons in your assay, you can theoretically achieve very high depths of over 16,000 reads per amplicon. At the other end of the scale, if you max out the assay with 96 samples all at once on the sequencer and you max out the assay to its limit of 384 amplicons, you'll only achieve a theoretical coverage of 256 reads per amplicon. So bear in mind, we want to achieve deep sequencing for our samples. So about the appropriate compromise, I guess, in our experiments is we've got we sequence 20 samples at a time on the MiSeq, and there's 212 amplicons in the assay, so we're probably achieving a theoretical read depth of around 2,000 reads per amplicon, which is enough for us to give us the sensitivity that we need. The next slide is just a run screen off MiSeq. This is the first. This is the sort of the QC screen at the end of a run, which shows you some important information that you can use to assess the success of the run. This shows in the top right-hand corner the histogram of showing read quality, and there's a line there at 75%. At Q30, which shows that in this run, 82.5% of reads were greater than Q30, which is impressively better than the spec for this instrument of 75%. You can also see, it's not shown on the screen, but I've written it underneath, that we've got a cluster density of 842,000 clusters per square millimetre, which is a really good cluster density for an experiment of this type. The spec for MiSeq ranges from 750 to 850. So one of the things that you can look at in a run is the amount of reads per sample. So this just shows the first 19 samples out of our beta test experiment. And we do see some variation in the read depth across samples. It's greater than you'd expect. The kit includes a normalization procedure, which should see each sample contributing, or the range of the variation between samples is no greater than twofold. Just bear in mind that we've been using FFPE material, so we don't have very good control over the state of this material, and particularly with samples 9 and 10, we put in a suboptimal amount of material, and that's why we're getting low reads. But still, there's enough reads there to find mutations. So the other thing we can do is look at Amplicons. Okay, so we're missing a graph there. This is a coverage plot of that experiment. It just shows you that the percentage of amplicons that are read at the depth shown on the x-axis. So, for instance, that 97% of amplicons were covered with a depth of at least 200-fold, and 89% were covered with a depth of at least 500-fold in this experiment. That's pretty good. So just a summary of that first beta test of TruSeq custom Amplicon. In the first sequencing run, we sequenced 19 samples. There are a total of 4,028 Amplicons. The average coverage per Amplicon was 2,600 reads. It's fairly high standard deviation, but it's a fairly wide, low normal distribution that we see with these reads. And 99% of reads with coverage greater than 50, and so on. Okay, so just wanted to show you some sensitivity data from the mixed cell lines. This is looking at some of those samples in the integrative genome viewer from the Broad, and this is just showing that one of the cell lines that we prepared, which had a theoretical abundance of mutation at 1.25%, we detected it in pretty much exactly 1% of reads from that Amplicon. And there's another mutation in that cell line, and we detected that in a similar proportion of reads. So in total, of that whole experiment, we had 33 known mutations out of the 88 FFPE samples, and we detected them all, so 100% accuracy compared to Sanger sequencing. So these experiments give us confidence that this assay is accurate. We've got a number of replicates in there. They're all in agreement. They give us confidence that the assay is reproducible. The cell line dilution suggests that the assay is highly sensitive. In using this assay in looking at clinical samples, we feel it's important to maintain a handle on these parameters. It's important to have quantitative controls. It is a quantitative assay, unlike Sanger sequencing. And so I think it's important that we include controls of known abundance in each run. And we've been talking with the Horizon Discovery company in the UK who provide genetically defined standards with precise allelic abundance, and we're going to be trialling those in our next experiments to see if they're useful controls in keeping an eye on the sensitivity of the assay. So now Cancer 2015 is our big project here at Peter Mac over the next few years, and I just wanted to talk a little bit about what this study involves. We're going to be doing 10,000 tumour profiles over the next 3.5 years. We started doing it, recruiting patients late 2011. We're up to about 150 at the moment. So the primary aims of this study are really to get a handle on variations in cancer care and survival and quality of life and health cost issues in the Victorian population. And particularly it's about mapping the prevalence of cancer mutations across the population without regard to the cancer pathology. And we want to be able to identify the true prevalence of predictive cancer genes in the general population. But the secondary aims are about facilitating the development of the infrastructure that's needed to provide the molecular pathology on this scale. And so Peter Mac is partnering with several companies to provide automation to allow us to deliver these large volumes of tests in a clinically relevant time frame. In particular, we've got PerkinElmer on board with some exciting robotic solutions. Robotics and automation is generally something that this assay could benefit from. Just creating a library on 196-well plate uses up to 16 boxes of tips, and it's we think that having an automated post-PCR system will really facilitate the delivery of this assay in a relevant time frame. So with the preliminary samples that have come in, we've decided to do a pilot study to just assess the viability of using the types of tumour material that we're getting delivered to us. We receive blocks or slides from a number of pathology providers throughout our state. We also get bloods, and so we've undertaken a pilot on the first 39 samples that have come in. We've also got access to the matching bloods. These 39 samples have been totally unselected for in terms of tumour type, tumour quality, or DNA yield. We wanted to see whether they would be universally suitable for Nextgen sequencing. So we see here the overall success rate is that we've found mutations in a little under half of the 39 tumours. Of the 20 that were negative, In fact, we'll see later on that over half of them failed because of low DNA quality. So we can easily conclude that some sort of upfront sample QC is necessary to get the maximum benefit out of this assay. This is a histogram showing an ordered series of Amplicons, and the x-axis labels don't show all of the Amplicons, but it really shows you that there's a range of read depths in this assay, and this is something that you should expect with an Amplicon library, because all these Amplicons are being generated in a single tube, and there's competition going on. So some Amplicons work better than others. The dashed line at around 10,000 is approximately our threshold for accepting a read. So later on you'll see that we've decided that, and it's a reasonably arbitrary decision, that we need to have at least 100 reads of an Amplicon before we are happy to accept a mutation call on that Amplicon. So over an experiment of about 96 samples, that's set at about just under the 10,000 line. So I think it's very important, particularly in these lower reads down here, Amplicons that generate a small amount of reads, that we have a quantitative standards to measure the accuracy of the experiment. So this shows you the mean depth of coverage per sample, and we can see in the left-hand side of this histogram here, these are all the FFPE samples, and then in the middle, change colour. These ones are the bloods, the matching bloods, and there's a few more FFPEs just here. And then these are some controls that we included, some of our own mixed cell lines. So you can see that clearly the FFPE material, without any upfront QC, is reasonably variable in quality. And you can see the ones that have failed. And we expect if we'd done an upfront QC, which is provided in the QC custom, the TruSeq custom Amplicon kit, that these samples would have been rejected and not put on the assay. Nonetheless, we went ahead and just had a look at correlation between read depth and the amount of input material that we were able to put into the assay. This is based on qubit concentrations, and so we can see that there's a strong correlation between sort of an input amount, total amount of DNA less than, say, 50 nanograms and low read depth. So that's these samples down here. And if you can get 250 nanograms into the assay, you generally get a very good read depth. There are some outliers, and we're interested as to why some of these ones down here, which have apparently low DNA input based on qubit, have given really high yields. That's of interest to us, and we're investigating those at the moment. What we hope is that the automation that we've got planned in the DNA extraction area will improve the quality of our material and allow us to get higher yields. At least it should facilitate our ability to post-QC to go in and re-extract with higher input amounts of tissue. So we're hopeful that we'll be able to drive up the quality and quantity of our material for testing. This just shows another view of that data, that of the 11 samples that had Amplicons with reads less than 100, that 10 of those had really low DNA input levels, below 50 nanograms. Only one, this one in the middle, K24, which interestingly was a lung sample, traditionally associated with poor quality, low yields of DNA, had high amounts. And you can look at it the other way, and if we show the samples that had a low amount of DNA, and there's 12 here, 8 of those had really low reads, which is really what that bar graph was showing, the scatter plot was showing earlier. Okay, so just a little bit about our results. This is a sort of a distribution of mutations that we've found in the main tumor types. And you know, it's gratifying. We found mutations in 22 genes, and they're pretty much the genes that are commonly mutated in cancer, and notably P53 and PIK3CA are well represented. Just some examples of particular cases. There's a cervical cancer here where there's clearly a driving KRAS mutation. There's a sarcoma that's got a really high proportion of a P53 mutation there. There's a lung with, interestingly, a BRAF mutation. That's not something that we'd traditionally test for in lung here at Peter Mac. And the bottom one is another lung, PIK3CA. And we can see the variant frequencies on the right-hand side are all range in the ability to detect in the abundance of those mutations. All these mutations are going to be confirmed by orthogonal techniques such as Sanger sequencing, and that's going to be the case throughout the Cancer 2015 program. One clinically useful result from this pilot study has been our single cancer of unknown primary. We identified a PIK3CA mutation, GLU545 lice, in about 33% of reads, whereas it wasn't seen at all in any of the reads from blood. This is a view of that mutation in the IGV viewer, showing a stack up of the A's versus the G's. And if we look at that mutation in the COSMIC database, we can see that it's a well-recognised mutation in all types of cancer. It's one of the, if not the most common mutation in PIK3CA. And you can further drill down into COSMIC and find that it's found in a number of different tumour types. Breast, large intestine, urinary tract are amongst the most common. And so we could hazard a guess as to what the primary tumour was in this individual. But I guess the point about doing this type of testing is that it doesn't really matter what the primary tumour is. The most important thing is that we've identified a driving mutation, and in this case it's an actionable mutation because there's drugs in clinical trials which are able to inhibit this tyrosine kinase. That's not something that until now has been easily done here at Peter Mac, so it's quite an exciting result for us. So I guess in terms of a progress summary with our cancer profiling, we're really confident that deep sequencing of multiplex Amplicon libraries generated from FFPE tissue is sensitive enough, it's reproducible enough, and it's accurate enough for us to use this type of technology, TruSeq custom Amplicon and the MiSeq platform as a basis for tumour profiling going forward. We're cognizant of the fact that sample quality is a critical issue in the success of these types of assays. It's vitally important that you have upfront QC, and it's also important that you have internal controls that allow you to look at the sensitivity assays, particularly around that lower threshold of detection. Importantly, to be able to deliver this type of technology in a clinically relevant time frame on a consistent basis, we believe we need to automate all phases of the analytical process, and we're undertaking that right now. I'd just like to thank a lot of people here at Peter Mac who've contributed to our success so far and continuing to work on the development of these programs. Trent Bosma is the scientist that's been doing most of the work on TruSeq custom Amplicon. I'd like to acknowledge Cliff Meldrum, who's now at the Hunter New England Health in New South Wales. Cliff was the driving force behind Nextgen sequencing and clinical pathology here at Peter Mac. I'd also like to acknowledge Jason Ellel, who's our bioinformatician, who's done fantastic work at creating a custom bioinformatics pipeline for Amplicon sequencing, which is what we've used to analyse all our data so far. And at Illumina, I really want to pay tribute to Derek Campbell, who was our application expert here in Victoria for a number of years. Derek's now returned to San Diego, and Derek installed our instrument and gave us a lot of help in the initial phases of TruSeq custom Amplicon. So, Derek, I know you're listening. We miss you, buddy. And that's it from me. Thank you very much.
All right, Dr. Fellows, we've got a number of questions that have been coming through on the chat page, so why don't we just try and go through these one by one. The first question, I think, had reference to some verbiage that you had on a slide, basically indicating that samples are genetically heterogeneous. The question was, should that read that they were phenotypically heterogeneous, and the follow-on to that was, are there no mutations in the normal cells within a patient with a genetically based cancer?
Are there no mutations in the normal cells of a patient? No, I wouldn't. I mean, I wouldn't go as far as to say that. It's very difficult to know what the mutation sort of load is in cells that are not sort of reached the phenotype of cancer, if you like. So I think what I understand about cancer is that there's an evolution and an accumulation of mutations over time. So very early on in a person's career, they probably start accumulating mutations which go on to be important in the final cancer genotype or phenotype. But at the moment they're out of our reach. Really, we can't detect them. So I think it's true to say that a cancer is genetically heterogeneous, that if you could look at individual cells in a tumour, and there's been recent experiments that show this, if you do multiple biopsies and look at multiple tumours from a metastatic tumour, that the mutational profile is different in different parts of the tumour. So it may be that they're also phenotypically heterogeneous, but they're certainly genetically heterogeneous.
Okay, great. Thank you. Another question, this is relating to your follow-on work here for the Cancer 2015 project. The question was, will the results be submitted into public repositories such as the TCGA project here in the U.S. and abroad, I guess?
Yeah, ultimately these results will be submitted to some sort of public repository, yes. Okay,
great. Thank you. The next question has to do with data analysis, and so I'm assuming that the question states, what data analysis software is used in this lab, and is it third-party software? So maybe if you could kind of clarify how you use the MiSeq reporter software and any other kind of informatics software that was used to derive the data here.
Sure. So there's a great deal of choice in for people that are wanting to do Amplicon sequencing as to how they do the bioinformatics. Certainly Illumina provides some really competent bioinformatics solutions with MiSeq, and but we've got a really solid bioinformatics group here at Peter Mac, and we've chosen to use open source programs such as gatk suite from the Broad, and we've developed our own custom pipeline to do our bioinformatic analysis, and we're pretty confident that that's the best way for us to go. Other people, there's a number of third-party programs available which are very good, and we haven't got any experience with those.
Maybe just a follow-on question there. How did you find the setup or the interface between the outputs using kind of a third-party software in gatk. How did you find the interfacing between the MiSeq output file and gatk? Was that challenging?
No, it's seamless.
Okay.
Yep.
Let's see. You have another question. I think this is a little bit specific to Australia here. So the question states, what do you see the steps required to offer this in a commercial diagnostic setting rather than research only, and specifically what would be required to make this happen in Australia?
Sure. So the regulatory framework in Australia is somewhat similar to in the United States in that clinical laboratories are accredited to ISO 15189 standard, and this type of assay would need to pass that sort of that sort of interrogation, the same as, for instance, a Sanger sequencing assay. And so the key really is to understand the analytical performance of the assay and to thoroughly validate it on blinded samples against a gold standard method. And these are when we do introduce the Nextgen sequencing assay into clinical use here in Australia, those are the steps that we will take. We will validate this assay to the same level as we would a non-NGS assay.
Great, thank you. Looks like maybe coming back a little bit to the performance of the assay. A question came in: are poor performing Amplicons consistently poor across different runs and sample types?
Yes, that's a good question. Yeah, we've done a few runs now, and we do see that some Amplicons, the same Amplicon is consistently poor. And so I think we have to bear in mind that the TruSeq custom Amplicon cancer panel is essentially, in the iteration that we're working with at the moment, is essentially an in silico design. It's not been optimized in a wet lab in any way. And there's a good amount of opportunity to improve the performance of some of those Amplicons by redesigning the primers, by making the primers a greater proportion of the primer pool. I guess there's lots of strategies available to the designers to do that. So, but yes, it's a consistent profile from sample to sample and from run to run.
So another question came in terms of, you mentioned earlier in the talk about kind of QCing the samples, in particular for the quality and quantity that's required. The question was, do you have any suggestions as to what you'd put up front for QC for that particular purpose?
Well, certainly, as a lab that's going to be doing a lot of samples, we would think that the QC step needs to be reasonably quick and reasonably cheap. And so the product that's provided with TruSeq Custom Amplicon is a good one. It's a quantitative PCR. It's designed to give you an idea of the performance of the functional performance of your DNA in an assay which is trying to amplify approximately 200 base pair products. And so I would think qPCR would be the obvious choice for that.
Let's see. It looks like we've got one other question on the assay itself. So I can go ahead and take this one. How many genes do these 212 Amplicons cover? Basically, the cancer panel will actually be hitting 48 genes using 212 Amplicons to cover all of that. Certainly, there's more information for people that want information on that at Illumina.com, and they can just search for the cancer panel straight off the web banner that's there. It'll take you straight to the information sheet and some of the other functional information about the assay itself. And I think we just got maybe one more question here. Actually, a couple. So what do you use as the minimum number of mutation reads to accept the presence of a somatic mutation in the sample, and how did you come to establish that threshold?
Yeah, so we have an arbitrary threshold of 100 reads of any sort of an amplicon. So if our pipeline says that there's a mutation present in an amplicon, the first thing we look at the moment is the total number of reads of that amplicon. If it's less than 100, at the moment we're not calling that as a valid mutation call. When it's over 100, I guess the question is, how many variant reads would we accept as being real? So, from I haven't got an answer to that from our experience, but I have had this discussion with other people doing Nextgen sequencing, and it's probably about 10 is probably a really safe number. So if you, or even maybe 7 or 8, if you see less than 7 or 8, you could be looking at some sort of, let's say, it could be an artifact of the sequencing chemistry or the history of preparation of the sample, the FFPE. But I think sort of a level of about 7 or 8 is regarded as a pretty safe bet that that's a real variant.
So one other question, and this is more maybe just to get your opinion on this. The benefit, if any, to using longer reads, specifically going after FFPE tissues. I guess trying to get to the heavily fragmented nature of these tissues and what benefit, if any, long reads would have in them.
Yeah, limited benefit really for FFPE. For us, we do have a program to introduce Nextgen into germline cancer genetics as well, for familial cancer, and that's really where it's going to have a benefit. But yeah, no, the question is right. Going to 250 reads is not going to bring much to the analysis of somatic tumours. But other upgrades that are coming through on MiSeq that's going to just produce more data. Even at 150 base pair reads, we're going to see more data collection by the end of this year. So that's going to be a good thing. But the long read chemistry won't really be much useful for FFPE.
Yep. I think, let's see, do we have any other questions here? So I can take this one here. So there's a question about the degree of freedom of primers for different genes or amplicons. So what I'd like to point out in reference to this question is that what Dr. Fellows is talking about here today is a fixed panel that you can buy directly from Illumina. However, the biochemistry that's at the heart of the assay does allow for the ability to customize specific sets of genes, or that you can go ahead and design in silico through the myillumina.com or web portal, and then you could go ahead and iterate your designs there, look at them, and go ahead and order those. So the fixed panel obviously is fixed, but you can certainly take the bed file and be able to load up and add content if you so choose. One more question for you here. It looks like, have you estimated the false positive rate? And if you had any, what type of positions or calls were they?
We haven't done that yet. We need to do a lot of validation on Sanger to work that out. So it's certainly an important thing to do. I think we'll find that CpG dinucleotides are probably likely to generate false positives through the modification of DNA in the FFPE, but that's something that we've yet to do.
Okay. Another question regarding, you'd mentioned that you found kind of 20 samples, kind of the sweet spot for this particular assay. And so just kind of question, I think, more about the availability of samples and, maybe getting your thoughts on that.
Availability of samples?
In terms of batching samples.
Yeah, I mean, well, we've got plenty of samples. I don't think, we would prefer to do bigger batches. And as as the ability of MiSeq to collect data increases towards the end of the year, we will do bigger batches because we've got plenty of samples.
Great. I think we're just about at time here. So last question was around if you noticed any kind of specific error pockets, certain Amplicons or anything unique about the error profile of the assay itself.
Not the assay, but clearly, there's certain genes are commonly mutated, but I couldn't comment on whether the assay is biased towards certain mutations or not. At this stage, I haven't got a feel for that. Okay,
great, thank you. So I think that we've kind of run through the gamut of questions here in the chat window. So I'd definitely like to extend my gratitude here on behalf of Illumina and everybody listening to a great presentation today, and also to point out that the presentation will be available sometime next week on the webinar section of the Illumina.com webpage. With that, I'd like to thank everybody for their time today and pass it over to Dana.
This ends our formal presentation. You may now disconnect.